All right, what's going on, guys? Today we're going to be talking about proteins and amino acids. So let's get into it. All right, guys, so when we talk about proteins, the first thing we should really pay attention to is what is the function of proteins in the cell? Why do they even matter, right? So there's four really big functions that I want you guys to remember. The first of which is proteins are enzymes, right? So all enzymes are proteins, and enzymes are really important in that they speed up the reactions within the cell to make things occur within a time frame which is actually useful to the cell, okay? All right, so the next function is a structural function. And this just means that proteins give the cells their shape, their structural integrity. They sort of act as a scaffolding for the cell, okay? And then we have a mechanical function, or you could even say like a motor function. And this just means that, proteins allow the cell to create movement, to contract in the case of muscle cells, and also to move things around the cell, okay? All right, and then the last function we need to talk about is signaling. And sometimes they even call this biosignaling, right? It just means that the cell is able to respond to different signals outside of the cell, whether that be, like signaling molecules from other cells or direct interaction with other cells. And this is really important for things like how your cells respond to hormones, how your cells respond to inflammation. It's really important in the immune system as well. Okay. All right, so now let's talk about the structure of proteins. All right, so really basic, guys, really basic. Proteins are just these things called amino acids linked together in these long chains, right? So you can think about this diagram here. Each one of these circles represents an amino acid, and then the lines in between them just represent chemical bonds between the amino acids. All right, so this is the basic structure of every single protein, okay? All right, and then really quick before moving on, I want to talk about where protein actually comes from. And you'll hear about this a lot. They say DNA is used to make mRNA, is used to make protein. All that means is just the DNA tells the cell in what sequence to put the amino acids to make different proteins. All right, and then where do we actually get those amino acids from? They actually come from the food you eat, right? So from the diet, specifically the protein in the diet, you'll take that, you'll digest it, you'll break it down into individual amino acids, which you'll absorb, and then your cell will take those amino acids and put it together in its own sequence to make its own proteins. All right, so it's important for you guys to know a little bit more about amino acid structure and also how their bonds form. Right, because teachers love to put this on the exam. So let's take a look at this. So looking at this amino acid here, let's focus on this carbon atom right in the middle here. And we're going to look at the different things that are bound to that carbon atom. All right, so over here on the left side, we have one nitrogen atom and two hydrogen atoms. This group together is called an amine group. All right, so the first group that's bound to the central carbon is an amine group. All right, now looking over here on the right side, we have one carbon atom, right, which has two bonds to an oxygen atom, and then it's also bound to an OH group, okay, which we also call a hydroxyl group. This group of atoms together is called a carboxyl group, and you might also hear it be called a carboxylic acid, right? So that's the second group that's bound to the central carbon. Then we just have a single hydrogen atom, okay? That's simple. Then up top here, we have the letter R, right? So what does that mean? The R, this is representative of what's called the R group, okay? And this could be many different combinations of atoms, right? Many different types of groups. And whatever's up here in place of R determines the identity of the amino acid. So you can think of this R, it's just a wild card. It's just a variable that you can put other things into to make other amino acids, okay? All right, so when two amino acids are coming together to form part of the protein chain, we call this a peptide bond formation. And this occurs in the ribosome, right? So if you remember that protein synthesis occurs in the ribosome, this is what's actually going on in the ribosome. All right, so we could see that we have one amino acid on the left here and one amino acid on the right. And we line them up so that the amine group that we just talked about is right next to the carboxyl group. And then this OH group over here on the left and this hydrogen on the right are going to come together and combine to form H2O. And that will just leave. It will leave the reaction, right? So it's an actual product of the reaction. And then, occurring in the same reaction, this carbon from the carboxylic acid will become bound to this nitrogen from the amine group, okay? And then this bond between them is called a peptide bond. And then we could continue adding more and more amino acids to either side down here with the same reaction, okay? And we keep building and building on the chain until we formed a complete protein. And because we're forming a bunch of these peptide bonds, you know what we actually call the final protein? We call it a polypeptide, which is the same thing as a protein. So anytime you hear polypeptide, that just means protein, all right? All right, so we talked a little bit about protein structure. We said that proteins were just sequences, chains of amino acids put together with peptide bonds, and that is true, but that's really only just primary protein structure. There's different higher levels of structure that we need to talk about. Right, so again, primary protein structure is just the sequence of amino acids within the protein. It's just the order that the amino acids are in. All right, then we got secondary protein structure, and this results from hydrogen bonding. So just remember that. Secondary protein structure is the result of hydrogen bonding, and it creates these different shapes within the protein. One of these is called an alpha helix, and this is the one that sort of looks like a ribbon being twirled around in a circle, right? And it's actually a very similar structure to DNA. The other structure is a beta-pleated sheet, and you can think of this as being like a zigzagged flat sheet. And multiple of these different structures can be within a single protein. So you could have, like 3 alpha helices and 2 beta-pleated sheets within a single protein, or any other different combination. All right, then we have the tertiary structure, and this is the overall 3-dimensional structure of a single polypeptide chain. And it results from the R groups of the amino acids interacting with the water in the cell. So you know that some of the R groups like to be around water, and other R groups don't like to be around water. So the R groups that do like to be around water will orient themselves to the outside of the protein, and the R groups that don't want to be around water will orient themselves to the inside of the protein. And the result of that is the protein gets twisted around and shaped into a new 3-dimensional shape. So the way that these R groups interact with the water, we call those hydrophobic and hydrophilic interactions. So hydrophobic describes the R groups that do not like to be around water, and hydrophilic describes the R groups that do like to be around water. And if you really want to know why the R groups behave, why they do, it's just based upon the chemical properties of the R groups, which is determined by the identity of the R group. All right, so the last structure we have to talk about is quaternary structure, and this is when 2 different polypeptide chains come together to form one complete functional protein. So 2 different protein chains with different amino acid sequences coming together to form one protein. And we can name these proteins based upon how many different types of polypeptides are involved. All right, so if we have a protein made up of multiple copies of one polypeptide sequence, we would call that a monomeric protein. All right, and if we have a protein which contains 2 different polypeptides, we call that a dimeric protein. 3 different polypeptides would give us a trimeric protein. 4 different types of polypeptides would give us a tetrameric protein, and you could keep on going. Alright guys, I hope you liked the video. I hope it was helpful. If it was helpful, please leave a comment below telling me what kinds of videos you guys would like to see in the future. And also, please subscribe to the channel. Stay sharp.